First question, what's the biggest single-cell analysis story of 2019?
Well, we think that one of the big things that we've seen happening in 2019, and it's been confirmed here, is CRISPR. CRISPR is really taking on a very large part of research, a lot of part of questions. So I think that's one of the key, let's say, driving forces from our side, where we've seen a lot of new things happening.
Fantastic. So what issues face the genomic and single-cell analysis researchers industry in 2019?
One of the things we've seen is that because of, let's keep on the subject of CRISPR, one of the things we've seen is it's opened up a whole new area where there's a lot of unknowns. And a lot of people are now looking for answers. And one of the things, for example, is the pathway that to get the CRISPR molecule to go into the nucleus and do its job. A lot of unknowns as how it does it, how it gets in there, and that is one of the big challenges that I've seen, at least in the questions that have been asked to us so far this year.
Fantastic. Number 3, what will 2020 hold for the single-cell industry?
Again, I'm focusing on CRISPR again because it's a hot topic. So we've got some people have heard of this new prime editing that's just come out, which basically means to be able to make much finer cuts within the DNA molecule. CRISPR comes in now in a modified form. Instead of cutting complete sections, it'll really be able to find pinpoint targets, and that is going to be a revolution insofar that it will really optimize the off-target effects. And the off-target effects really is the big problem in CRISPR therapies today. So yes, CRISPR does solve problems and solve diseases, but it can create others. And those are unknown. And that's one of the big questions that the industry now is trying to find answers on.
Fantastic. So what will be the biggest technology impact for 2020?
Okay, being a little bit maybe personal and selfish here, we believe that our technology, which is the Fluid FM direct delivery into the nucleus technology, will really help push this technology forward. Which means when you can put the Cas9 and whatever goes with it directly into the nucleus rather than having to go through the cytoplasm, it means that you know exactly what you want, what you have, and you put it where you want so it can do what it needs. When in the other technologies, we see that having to go through the cytoplasm, sometimes the DNA template can get damaged or modified, which means that when it gets into the nucleus, it may not actually do either anything or do the wrong thing. So this technology that we have, I think, is going to open lots of doors to be able to do things in a very, very different way and help progress CRISPR even better and faster to save lives and bring therapies to the market.
Fantastic to hear. So final question, what are the future challenges researchers are facing in this field? So the idea is then to take the theory that I've just mentioned and actually get some facts. And we have, Lin Wu today, I think in one of the, mentioned in one of the panel meetings that delivery was one of the concerns that they had. So yes, we think we can solve that, but now we have to prove it. So we have several partners with whom we're working. Unfortunately, I can't name them. I'd love to be able to, but I can't name them. But we have 2 or 3 partners we're working with to be able to make those proof of principle experiments to show that what we think can actually really happen, that the HDR efficiencies and everything that we've talked about can really improve things. And then that will really start getting going. So hopefully this time next year, we'll be back here with some new talks, and we'll be able to share these results with the community next year.